Welcome to Module 6, Decontamination and Disposal of Biohazardous Waste. What is disinfection as it relates to biohazardous materials? Disinfection is the destruction or removal of all pathogenic organisms or organisms capable of causing infection, not necessarily spores. What is decontamination? Decontamination is the use of chemical or physical means to remove, inactivate, or destroy biohazardous agents on a surface so agents are no longer infectious. Effective decontamination, disinfection, and disposal programs include routine disinfection of lab spaces, decontamination of biohazardous waste, and appropriate storage of biohazardous waste as it is accumulated. There are a variety of decontamination methods available. Whatever is selected needs to be appropriate for the following: the condition in which it is being used. For example, do not use bleach on stainless steel unless you have a plan for wiping residues off afterwards to prevent corrosion. The overall effectiveness in inactivating the agent. For example, boiling water does not inactivate endospores from spore-forming bacteria. Consider all local, state, and federal regulations regarding the requirements for decontaminating biohazardous waste. The link to the Wisconsin Infectious Waste Regulations is available in this module on Canvas. The method selected should also be safe, practical, and cost-effective. There are two means of decontamination: physical methods and chemical methods. Physical methods of inactivation include hot methods such as autoclaving, incineration, and a dry oven. Cold methods include refrigeration, freezing, or desiccation. Chemical methods involve the use of varying chemical compounds to inactivate biohazardous materials. These include alcohol, such as 70% ethanol, chlorine and chlorine compounds, such as bleach, aldehydes, including formaldehyde, glutaraldehyde, and orthophthaledehyde, ethylene oxide gas, hydrogen peroxide solution and vaporous hydrogen peroxide, iodophores, such as iodine, peracetic acid, sometimes in combination with hydrogen peroxide, phenolics, such as Lysol, and quaternary ammonium compounds or quats. Let's discuss biohazardous waste decontamination and disposal methods. Solid waste, such as plates, solid media, used disposable PPE, or any other solid materials that may be contaminated with biohazards, should be collected in labeled biohazard containers lined with orange or clear autoclave bags. The container must have a lid for storage. When 2/3 full, loosely close the bag and autoclave in a secondary container with a chemical indicator, and then place the cooled waste in a black trash bag and move it to the dumpster nearest your building. Plastic and glass pipettes should be collected in a box lined with an autoclave bag, either orange or clear, then autoclaved out. Place the autoclaved bag back in the cardboard box, seal it, and dispose of it in the dumpster near your building. Pipette tips can be collected in a small benchtop autoclavable biohazard bag and then added to the solid waste autoclave cycle in the larger bag and disposed of accordingly. Medical sharps, such as needles, syringes, razor blades, scalpels, etc., must be collected in an approved sharps container. Then the exterior of the container must be decontaminated and the container must be sealed prior to calling hazardous waste for sharps pickup. Do not put it in the trash. Liquid waste, such as liquid cultures, human blood and bodily fluids used for research, animal cells in tissues, and cell cultures must be decontaminated by an appropriate means, such as the autoclave or a chemical inactivation, and then may be disposed of down the drain to the sanitary sewer with copious amounts of water. Here are some reminders for safe sharps waste storage and disposal. If sharps must be used, select those that have the best controls in place to minimize the risk of injury. Keep a clearly labeled sharps disposal container nearby for disposal. Use safer medical devices, such as a self-sheathing needle or needleless system. Do not recap, break, bend, or cut needles after use. Sharps containers must be hard-sided with a cover. They do not have to be the red Sharps container. They can be a defaced container from non-toxic products, but it must seal shut with a lid to prevent accidental spilling if tipped and clearly labeled. Seal the container when it is 2/3 full and decontaminate the outside of the container prior to pickup. Do not overfill, push down to make more room, or leave it open for spilling. These are autoclave guidelines that are also available in the Canvas module. It can be used as an SOP posted by your autoclave. Feel free to download this and put it by your autoclave space for your group's use. Let's review where all of your hazardous waste should go. If it contains chemically hazardous waste, it must be handled by our hazardous waste program. Label the waste containers clearly. Contact the UWM Hazardous Waste Program for pickup. If it contains radioactive materials, it must be handled by the Radiation Safety Officer. Contact the Radiation Safety Officer for disposal. If your materials are potentially infectious materials, it is the responsibility of the principal investigator and their lab personnel to decontaminate their waste prior to disposal. After it has been appropriately decontaminated by an approved method, waste may be disposed of in the general trash stream except for sharps, which must be disposed of through our hazardous waste program. If the waste that needs to be disposed of contains both biohazards and chemical hazards, the chemical hazards always supersede the biological hazards. Treat it as a chemical hazard, and it must be picked up by hazardous waste. It is the lab's responsibility to move their trash from their labs to the dumpsters outside. Do not expect environmental services staff to pick up the trash in your laboratories. With reduced staffing and the increased risks of working with biohazardous materials in your laboratories, you need to be responsible for moving your trash to the dumpsters. Do not leave overflowing trash bins in the autoclave rooms. Decontaminated waste may be added to a black trash bag, sealed, and carried out to the dumpster. Clean up after yourselves. This is part of being a responsible lab personnel at UW-Milwaukee. Someone in environmental services is responsible for picking up trash. Would you want to expose them to anything? Additionally, the outside community and the environment will be exposed to whatever you place in the trash. Each research lab must have a standard operating procedure for their group of how their equipment is decontaminated, how waste is decontaminated prior to disposal, and for the autoclave use, if applicable. They are responsible for understanding and following their standard operating procedures. Do you have additional questions about waste decontamination and disposal? Contact the Biosafety Program Office at uwm-biosafety@uwm.edu with your questions. This is the end of Module 6, Decontamination and Disposal of Biohazardous Waste. Complete the quiz to move on to Module 7.